cellulose. And cellulose is very stiff, it's very firm, and it has a very strong structure. And part of that is because rotation is limited by sterics. And so here's another picture of cellulose. And we see here that this hydrogen bond is a linear hydrogen bond. And so it's a strong bond. It's more stable, and so it's less likely to rotate. And therefore cellulose has a very strong structure. And so here in amylose, and remember amylose is just one part of starch, you see that amylose has a bent hydrogen bond, and therefore it's not quite as strong, and we would expect that its structure wouldn't be as strong as cellulose. And here's dextrans, which is a sugar that you see in bacteria. So here we're going to introduce you to GAGs. And GAGs are glycose amino glycans. These are heteropolysaccharides, and they're repeating disaccharide units. And so there's 4 types. So we see them right there, 1, 2, 3, and 4. And notice that hyaluronate is composed of 50,000 repeating disaccharide units. So you would see 50,000 of these right here. So how many monosaccharides would you find? You would find 100,000. So hyaluronate is the largest GAG of the 4. And then you see that I circled heparin, and that's because heparin is going to be quite important in the next couple slides. And if you're taking notes, you'll want to note that heparin is it's a very negative molecule, and the negative charge is conferred by a sulfate group. And look at all these negative charges. So it's a very negative molecule. Just a quick side note, you'll want to do question 24. And this slide probably should have been shown a little bit earlier, but I earlier broke down starch, and I referenced it as being a homopolysaccharide, and then talked about amylose and amylopectin, and that it was the storage form of energy in plants. And you'll want to make a similar list for glycogen and cellulose. Note that chitin is the structural element of insects, and dextrans is the structural element of bacteria. And you see here, do question 7, 18, and 19. So here we're introducing you to a new structure called a glycoprotein. Sorry, this is a proteoglycan. Okay, and this is showing the protein right here, core protein, and this is showing a GAG. So chondroitin sulfate is a type of GAG, and so we are adding a GAG to a protein. And this is showing the repeating unit that we always add the GAG to. And so here are 2 types of GAGs, or sorry, 2 types of proteoglycans. You have a syndecan and a glypican. And so here's the core protein. So just on that earlier slide, here's the core protein. That's this red part. And then this wavy thing coming off of it is the GAG, chondroitin sulfate. And note which of these is a transmembrane unit, the syndecan, and the glypican is an external unit. And you see here that this has a cleavage site on it, and so these will both be cleaved, and they will become part of the extracellular matrix. So I mentioned earlier that heparin was going to be important, and one of the important characteristics of heparin is that it has this NS domain. So this thing right here You see it's just a portion of that GAG. And so the NS domain is highly negative, and so you would expect it to attract or alter certain other proteins that have a positive charge to them. And so here we see 4 ways that the NS domain can alter the structure of other proteins. And so we see here that this box protein is going to be attracted to the NS domain and bind, and when it binds, it's going to change conformation. So number 1 would be a conformational change. B, I like to think of this as tethering. The NS domain will help bring 2 proteins together and tether them together, help them to interact with each other. Here we see that the NS domain is helping to bring the ligand to the receptor. And over here we see that we're localizing an enzyme at this NS domain. So if we were to need an enzyme near the surface of the membrane, we can store the membrane, or we can store the enzyme here ahead of time so that we can have quick access to that enzyme. Here we see a special type of proteoglycan. This is called a proteoglycan aggregate. And so here we see that this core right here is hyaluronin. And remember that hyaluronin is the biggest GAG. So here you have GAGs. And then coming off of here, these things are just proteoglycans. And so it's a big GAG with a bunch of proteoglycans coming off of it.